All right, so in this video, we're going to be looking at DNA replication, which is D1.1, and we're going to be talking about how new DNA is produced, and we want to be thinking about continuity and change, which means, how are we keeping DNA structure more or less the same, but also simultaneously, how are we making copies of it so we can replicate cells? So DNA replication is a pretty complicated process. If you're SL, you don't need to know too much about it. DNA replication is for copying itself, and it's going to use one strand as a template, and the other strand is going to be copied onto it. This is called semi-conservative replication. And complementary base pairing is going to be happening, which means A's and T's are going to join together, and G's and C's are going to join together. And so there's two enzymes that we need to be aware of for this process for SL level, and that is helicase. That's this enzyme right here. It's going to unwind the double helix, so it's actually cutting the hydrogen bonds between the bases so that the strands can separate. And then the second one is DNA polymerase, and DNA polymerase is right here. So it's going to bring free nucleotides into the vicinity of the binding region. And if they can make a complementary base pair with their pair base, then they will form hydrogen bonds, and then the DNA polymerase will seal together the phosphate and the sugar. And this purple band is the phosphate sugar band, and then the multicolored are the bases. So it's bringing the free nucleotides and then linking them together. And in order for DNA replication to occur, there's a wide range of components called the replisome that are needed for this process. Helicase and DNA polymerase are two components of the replisome, and they're all that we will look at for SL level. So when we are looking at DNA, we could also use DNA for DNA profiling. And in order to do that, we're going to use something called PCR, which stands for polymerase chain reaction. You've probably heard of the acronym PCR for, have you had a PCR test related to COVID? And so what's happening with PCR is that it is making more replicates of the DNA so that they can be tested, and there's sufficient DNA for the test to come back positive. And so what's happening here is DNA samples, primers, nucleotides, and something called Taq polymerase are all added into a thermal cycler. And that thermal cycler is going to denature the hydrogen bond by putting it up to 95 degrees. So if this were normal human DNA polymerase, that would denature that enzyme and it wouldn't work anymore. But luckily, Taq polymerase actually comes from a bacteria that lives in hydrothermal vents. So their DNA polymerase works at very, very high temperatures. And so that's why this is used in the thermal cycler. So denaturing is sometimes called melting. It's going to separate those hydrogen bonds. And then annealing is where Taq polymerase is going to add on the bases and join up the phosphate sugar backbone. And then extension is where the full strand of DNA has been completed, and that's sometimes called elongation. And then after PCR has been done, gel electrophoresis would then be utilized to see if the DNA fragments match a sample sample of DNA that has been collected. And so we'll look at that more on the next slide. But what's going on there is the DNA segments are loaded into these little wells in the gel plate, and electricity is applied. And when the electricity is applied, the negative electrons are going to move towards the positive terminal, and it's going to move the segments of DNA. And so if the segments of DNA they only move a short distance, that means they're quite heavy, which means they're quite big. And if they move a longer distance, then those fragments are quite a bit smaller. And so we're looking at, do these fragments match the fragments of the suspect or the person that we're trying to test? And so when we are looking here at a COVID test, they had to jam that swab straight up your nose, and they're going to extract viral DNA from that. Then using something called reverse transcriptase, they're going to make DNA from that. and they're going to run the PCR cycler 35 times. So there's sufficient DNA to test, and they're going to fluorescently tag any DNA, any viral DNA that's present. And if the computer detects that a certain color is reached, then you have a positive COVID test. When we're looking at testing paternity, for example, this mother and child, so the child has some segments of DNA. These are called short tandem repeats. These are sequences that repeat on certain chromosomes, and they're unique to the individual. And so, for example, a GAA is a sequence that can repeat on chromosome 18, and it might happen 7 to 27 times. So I might have 7 repeats, but you might have 27 repeats. TAGA repeats on chromosome 5, and it can repeat 6 to 15 times. And in order to do a paternity test, they are going to run their PCR at least 13 times so that they have sufficient DNA to test different potential fathers. And so the child's going to get some of their short tandem repeats from their mom and then some from their dad. And they're not going to have exactly the moms or exactly the dads. And so if we're looking at this child who is the father in this case, well, this child doesn't have that mom's stuff, but it is missing an STR there. And dad 2 and dad 3 are both missing. So that could be an indicator. Similarly, this gene segment doesn't exist in mom, but does exist in dad 2 and dad 3. So dad 2 and dad 3 are strong contenders already. This one is in mom. These 2 are missing here. Dad 3 is missing some genes in there. So that could imply it could be dad 3. But dad 3 has this guy, this STR, and kid does not. So these are from mom. Then there's a space. Dad 3 also has a space here. And looking at these ones here, it looks like this one is present in dad 2 and dad 3, but this one is the one that also, it shows up in dad 1 and dad 3. So dad 3 is the child's father. And these strands would be separated using gel electrophoresis, and the pattern is unique to each individual. So you can see that the child doesn't have the exact same strands as mom, nor does it have the exact strands as dad. So to summarize, for SL students, DNA replication is copying itself from a DNA template using DNA polymerase and helicase. And helicase is going to unzip the DNA, breaking the hydrogen bonds. DNA polymerase is going to bind nucleotides together, forming covalent bonds between the phosphate and sugar. Transcription is RNA being made from DNA templates, which we're going to look at in a further video. And then translation is making proteins in ribosomes from an RNA strand. But this video was looking at replication, DNA copying itself. PCR makes copies of DNA, and then gel electrophoresis separates the strands to see if we can find a match with an individual. So for SL, you can stop there. HL, you should keep watching because we need to talk about a number of different other factors going on in the replosome. So DNA replication, nucleotides are added to the 3' end because the OH group on the carbon there at the 3' end can bond. It's going to make a covalent bond with the phosphate group. And so in this case, on the leading strand, so helicase is going to bust open the hydrogen bonds. And then one enzyme that we need to know is topoisomerase, sometimes called gyrase. Topoisomerase is going to, when the hydrogen bonds get cut here, it creates a bit of torsion and twisting on the remaining double helix. And so topoisomerase reduces that torsion. When the strands are open for bonding, we have the leading strand and the lagging strand, and these replicate differently. So the leading strand and lagging strand are both going to replicate on the 3' end. In this case here, on the leading strand, this is the 3' end, but actually on this side here, on the lagging strand, this is the 3' end. So we're going to have to do things a little bit differently on the lagging strand. So continuous replication on the leading strand goes towards the replication fork, but it's discontinuous on the lagging strand. And these discontinuous pieces are going to be called Okazaki fragments. the replication process is slower on the lagging strand because it's constantly restarting replication with DNA polymerase. And there's a few other components going on here that we need to know about too. So we've already talked about topoisomerase, which is sometimes called gyrase also. And what we do need to know here is DNA primase. So DNA primase is going to add an RNA primer segment. That's this little blue one here. And that's going to allow the replication process to start. And it's going to start going this way, boop, boop, boop. And DNA polymerase 3, in SL, we just called it DNA polymerase, but there's two different types that we need to know about, DNA polymerase 3 and 1. DNA polymerase 3 is going to add the nucleotides and bind them together. DNA polymerase 3 is also going to proofread for any mistakes, and it's going to remove mismatched bases. And once the fragment has been made, then what ends up happening is it seals onto the next fragment using something called ligase. Ligase attaches the Okazaki fragments together using covalent bonding. And additionally, just before ligase is in action, DNA polymerase 1 has to remove the RNA primer from between the Okazaki fragments. And so you would have an Okazaki fragment here with an RNA primer. That primer needs to come out of there, and in order for both of these to join together. And then ligase will attach the Okazaki fragments together. All right, so that is all for this video. Thank you for watching.